Hi, Mike. It was good to meet you yesterday. So, as promised, we're just going to have a wee look at your soil here. So, let's just have a quick scan and then I'll just give you a kind of rundown of what we're looking at and why it might be interesting. So, basically, we're looking for all these functional groups in the soil. All these, excuse me, my voice is going in an opportune moment. All these trophic levels of life, basically, that we're looking to see in soil. If we're basically, at its most fundamental level, if we're seeing all these trophic levels, then generally speaking, the soil is, what a human would regard as healthy or maybe productive would be a better word actually for it. So I'm sure you've heard of all this, but sort of optimal growing conditions for vegetables, or at least most vegetables, if they're annual vegetables, this doesn't really count for most perennials, is a fungal to bacterial ratio of 1:1 in terms of the biomass, in terms of their raw weight. There's obviously more detail to it than that because, we're wanting to see lots of species diversity. So that's the other key point with soil, is we're wanting to see a huge diversity of life, so that no one species or trophic level can dominate over the others. That's when you start getting diseases and issues. Bacteria being one of the big functional groups, of course, fungi, of course, being the other one. And then we have kind of many of the predators, and they'd be starting at the smallest as the kind of protists, basically, the flagellates, amoebae, and ciliates. And then there's nematodes, as you're starting to scale up. And then, of course, all the macro arthropods, the earthworms, etc. Earthworms, interestingly, being carnivorous. They're predators that actually eat the bacteria growing on decaying matter. It's not the decaying matter they're actually digesting. Another just little, I'll try not to get too diverted here, but another interesting little bit of research that's come out recently is the rhizophagy cycle. It's definitely worth looking that up. James White and his research has kind of found that, and it turns out that all plants are actually predators and carnivores as well. So it feels like every time we basically don't have much of an idea of how plants and soil function, and as George Monbiot says, we don't have a clue really what soil is particularly. Some people regard it as a single entity, an organism. All the things living inside it would just be equivalent to its external stomach, the grasses and forbs that live on it kind of act as its own immune system to protect it, and you could look at it that way. I think that's quite a good way of looking at it, to be honest. But anyway, we're getting totally sidetracked here. So let's have a wee scan and actually tell you what's going on in this soil. So I'm just scanning. This is very low magnification, like times 40. I'm basically looking for arthropods and nematodes at this resolution. Nematodes you'll see because they're wriggling around, so they're quite easy to find. Nematodes get a bad rap because of root canker and root rot nematode, all those kind of things, but there's many different species of nematodes, many of them completely disinterested in your plant roots, and they're fundamental for nutrient cycling and population control of bacteria, fungi, keeping everything in check and balance. So kind of another fundamental principle is, where do plants get nutrition from? I'm sorry if this is like completely patronizing, you know all this stuff already, but you can just skip this. But plants get all their nutrition from the activity of microbiology, essentially the excretions of microbiology. So a very simple example would be protists coming along, and amoebae eats about 10,000 bacteria a day. Those bacteria have a concentration of nutrients within them. They've got those nutrients from using their enzymes to break down bedrock material. And those nutrients are in too high a concentration for the protist So it excretes the excess. And generally speaking, a lot of the activity is around the plant roots, the rhizosphere, and that's directly excreting into the plant roots the plant available nutrients that the plant requires to grow, all its macro and micro trace elements, its macronutrients. All of that comes from the activity of biology, unless of course you're adding synthetic fertilizers, etc. But yes, that's how they become made plant available. And of course, there's many other reasons, as you know, for having healthy soil. It allows for excellent water infiltration if you've got good structure. It allows for gas exchange, not only oxygen, because of course plant roots need oxygen in order to be able to function correctly, or many of them do, unless it's some sort of pioneer species or a rush or something like that. And yeah, so I'm not actually seeing any nematodes here, but let's just have a wee zoom in and see what we can see. So here, what we're actually looking at, so it's a bit more in focus for you. Generally speaking, the honey-colored little globules, so that thing in the center, it's kind of honey-colored, that's fulvic acid. And that's concentrated excretions made from the activity of microbiology. So as something is consumed and then excreted, it becomes concentrated. And then that leftover waste product is eaten by something else, and then something else, and then something else, and then something else. So it's passed through the body of many microorganisms. But it started as organic matter, and it's been concentrated over time. The darker, basically, the more humic acid levels it will have within it. And these are what you would call aggregates, really. So that's what gives, you know what an aggregate is. It's the crumb structure, basically, of a soil. And that is also the activity of microbiology, of course. They're like, it's good to think of soil as a coral reef, because that's exactly what it is. It's biologically engineered structure. That is what soil is. They've made it their home. It's the glues, the glomalins, that microbiology excrete that allow for them to bond together all the organic matter in the soil and create a home for themselves. It's a place of shelter and refuge. It's an edible home as well, of course. The more transparent little bits that you can see are little bits of silica, little bits of sand particles. So this thing right in the center that's kind of semi-transparent. You can see clay and silt particles. They're just very, very small, much smaller than the sand particles. So let's actually just zoom in even further. Maybe I'll catch some activity then. There we go. So now you can see a little bit of movement. Those are bacteria. So everything that's moving is alive. It's moving possibly under their own propulsion, possibly under Brownian motion. But they are, those are all bacteria, single-celled organisms. There's something like about, on average, 1 micrometer across for many of these cocci species. So we want diversity in everything. We're looking for many, many different shapes of bacteria. So yeah, I'm seeing cocci bacteria. What else am I seeing here? Looking for bacillus, like your classic rod-shaped bacteria. Don't see many of them. Can't see any of them, but we'll keep searching. But it's searching with bacteria, this. And generally that's what you can expect to see on a field that's been recently ploughed or tilled. sort of every time you plow or till a field, you lose about half of your fungal population, unfortunately. But you do get a growth response because of all the creatures that were killed basically instantly release all of their plant available nutrients into the soil, because the ones they were holding within their bodies. So you do get a plant response from a growth response from plants. So yeah, looking for fungi, definitely. Looking for fungi at this magnification, about 1 fungi per field of view, something like that. This is some sort of a mineral here. It's possibly a salt particle, actually. This thing in the middle that has 3 straight edges. There's a diatom there. That thing that looks a bit sort of like a inflated condom or something, tied up at both ends. That's a diatom. So that's what diatomaceous earth is made from. There's fungi. So let's try and get him like really good in focus. So you can tell it's a fungi not only from the color, but you can see that there's little dividing lines, little cross walls, septa. And generally speaking, we like to simplify things. We divide fungi into groups that are maybe mutualists, groups that are parasitic, and neutral groups. But it's also important to remember that any species can act as a parasite, and there's almost no obligate parasites in nature. So there's very few species that actually are always parasitic. They're reacting to the environment that they're in. So under these conditions, it appears that this fungi is doing well. You would judge it as a beneficial fungi. Probably an Ascomycetes. So it's a nice dark color. That's actually melanin, like the same stuff that your skin tans with, or like freckles, basically. That's what they actually use to protect themselves from UV. It's got evenly placed septa, and it looks to be quite wide. Generally speaking, the wider the better. So that's good. That's cool. We're looking for a lot more of them, though. Let's see what else we've got here. So plants to facilitate all the activity that you find in soil. Plants are pumping down anywhere from 15 to 17% of the energy that they get from photosynthesizing as sugars directly around their roots. They're pumping it out into the soil, and this is to excite and stimulate microbiology. In order to, there's a fungal spore right there in the center. I think it's all divided up. That's a fungal spore. In order to excite and feed microbiology, in order to get this nutrient cycling happening. So the other benefits that microbiology bring is that the more complex microbiology becomes, the more trophic groups we've got, the further ahead in plant succession the soil will be pushed. So when I say that we're trying to get the fungal to bacterial ratio at about 1-to-1 in terms of biomass, that will push plant succession into a point where most vegetables can dominate. Pioneering species that we call weeds, but are just really pioneering species that are generally speaking adapted to unhealthy soil, or you could look at them trying to heal soil. They will not be stimulated to germinate and grow when you get up to those kind of levels. So generally speaking, you see a massive reduction in weed growth if you can get your biomass up to that point. You're going to see healthier plants because they're able to access all the nutrients that they require, and they'll be able to fight off disease. And some people have done studies where if you're able to get a plant growing in its optimal soil conditions, and it has a coating of microbiology all over its roots, all over its leaves. They're not going to suffer from predation and pressure. They have a very fiery immune system that's going to be able to defend itself. So the classic example is like the Candide aphid. So when an aphid bites an incredibly healthy plant that's on top form, it's going to be able to pump sugars that will overwhelm the aphid's ability to osmosize them out. It's going to pump sugars directly into that aphid and candy it, kill it stone dead, which is quite amusing. So yeah, you see those sometimes on really healthy plants. The aphids look like they've got their proboscis stuck in the plant, but actually they're dead and they're just full of sugar. And the same goes for caterpillars. Same goes for grasshoppers, etc. They're able to reinforce their cell walls, their guard cells, and their cuticles to make it impossible for something to easily digest it. Slugs and snails are generally speaking attracted to dead or dying plants. So yeah, what's we got? So yeah, we can see like very rapid movement. I was wondering if that was an amoeba, but it's not. Flagellates look really drunk, so they're one of the Protus family, one of the predators. They'll feed primarily on bacteria, and they kind of look really drunk, and they fly around and they swim around in circles sometimes, and they bump off a bit of organic matter and head off in the other direction. Ciliates are like on a complete mission, and they go in a very straight line, and they know exactly where they're going, and they land on bits of organic matter often and hoover around them, hoovering the bacteria off. Other amazing things you can see, of course, are like nematode trapping fungus. I've seen them once before, so that was quite exciting. They make little lassos, grow little lassos at the sides of their body, and produce hormones to attract nematodes into the lasso. And then when the nematode is in, it sort of triggers a mechanism where it suddenly flushes the lasso full of water and traps the nematode, and then slowly digests it. And actually, those species actually prioritize root feeding nematodes. They'll preferentially eat them. This is another fungi here. You can see just tucked away. That's good. It's quite different in appearance. It's still fairly even in diameter. It's not nearly as dark coloured. The septa look to be less even. So yeah, we would rate that as maybe not parasitic, but maybe not quite as beneficial. So yeah, one of the ways that it might be worth, and I mean, I'm happy to come down and just kind of do this if you fancy doing a wee experiment. Maybe this looks like a nematode that's curled up here. Let's just see what you are. It looks like it's got the end, that pointy end is quite indicative of a nematode's tail, but I don't think that actually is a nematode. There's no you can see all their internal organs pumping away, so if it is a nematode, it's dead. Making compost, Berkeley compost, or maybe making a Johnson-Su bioreactor would be quite interesting. And inoculating your soil with a lot of biology, to try and, in an effort to try and reduce weed pressure. Yeah, be an interesting experiment. If you have like one small bed or something, try like a radically different management approach to it or something like that, and see what kind of results you get. It might be quite fascinating. There's another bit of fungus here. Very nice. So yeah, I mean, to me, I'm not seeing the sort of trophic levels that I would want to. There's another bit of fungus here, actually. It's quite good. Nice dark, rich colour. And because there appears, I mean, this isn't obviously like this isn't an assay at all, but it might give you some indication as to what's going on. Yeah, not seeing sort of all the predators particularly that I'd be want to seeing in healthy soil. Looks to be very bacterially dominated, and generally speaking, you can see that effect above ground. Maybe it floods easily, or actually it doesn't hold moisture very well. Or you have a huge amount of weed growth. A lot of weed pressure is an indicator of bacterially dominated soils. Or you have poor growth from specific plants. So there's obviously some plants that don't benefit from ecto or endomycorrhizal fungi and actually thrive, there's another bit of fungi there, actually thrive in conditions where the soil is bacterially dominated. So those would be like brassicas, lettuce, I believe, as well. Yeah. That's their kind of niche in nature. Even the cultivar ones that we grow. Yeah, interesting. So yeah, I think that might be, I don't know. I mean, if you're up for doing something, just a tiny little, you could just do 1 meter by 1 meter square. Something like that. And I don't know if you have seen anybody with a Johnson bioreactor or something. We're trying to do at Lauriston because we're kind of running into these same problems. We also get Caledonian horticulture compost, which is really rubbish, like we were discussing yesterday. Compost, in my opinion, it shouldn't just be bulk organic matter that's fermented. Those are not optimal conditions for a plant to grow in because it arrives like dust, has no structure in it whatsoever. So I don't think that should be, we really need to redefine legally what compost is. That's not compost. And somebody needs to start up a really good composting business so that, yeah, all these sort of market gardens and everything can benefit from it. But compost should be an inoculation of life, is really what it should be. But yeah, interesting. Big bits of fulvic acid you can see here. And there's lots of detail I missed out. I'm trying to just not be too specific about anything. But generally give you a brief overview what it looks like. Interesting. Yeah, so we'll call it there. And next time I'll probably come meet up with Maria at some point, so I can take some other samples by means of a comparison, because I only actually took one sample there. But that's always useful to know what to shoot for. So yeah, cheers, Mike, and thanks for letting me wander around and see the farm. It's really interesting. It looks like an amazing growing project. So yeah, hope you found that interesting. All right, cheers, bye.